master degree in preventive veterinary medicine and business administration. He worked in the university as a lecturer in the poultry disease, and he moved across to Agrocres Ross. Today is Epigenetic do Brazil, sorry. And he spent there around 10 years. Then he joined Seva in 2004, initially in Asia, then in France since January 2017 for the veterinary service group of Seva Animal Health in Asia. He is one of our main experts with 34 years of experience in the poultry sector. So I know that we will enjoy your session today, Dr. Paniagua, and the mic and the stage is yours.
Thank you. Assalamu alaikum. Good morning to everyone. It's really a pleasure to be here and to be able to share a little bit of what we learned about Newcastle disease here in Asia. Asia, as you know, is an area where Newcastle disease is very present, is endemic. And I just would like to share what is happening to producers, what producers are doing to try to reduce the losses associated to this disease. Okay, so let me take 1 second here to share my screen. Okay, I hope you can see it.
Yes, we see it, Marcelo.
Okay, that's perfect. So the topic for this morning is Newcastle disease, threats, and prevention strategies, as I mentioned. It's really an opportunity to share some experiences and to show to you how producers here in this part of the world are dealing with this important disease. So I would like to start this presentation just telling you a few points related to Newcastle disease. The first one, and the most important, is Newcastle is present everywhere. It has a worldwide distribution and is enzootic in several countries, including here in Asia and probably in Saudi Arabia as well. For your curiosity, only 16 countries, only 16 have never reported an ND outbreak. And all these 16 countries are small islands in the middle of the Pacific Ocean. So this disease is pretty much present everywhere. And the impact, both economic but also social impact, are huge. They are enormous. Just to mention 2 of them. The first one, in those poor countries like Bangladesh or some more African countries, this problem can create problems of food security. As this disease kills lots of birds, so it can really create problems related to food security. And on the other side, for countries like Brazil, my home country, Brazil exports lots of chickens, you know that. So if there is an ND outbreak, possibly there would be ban on exports. So there is lots of problems related to that goes beyond of the mortality in the farms, right? But let me show you the evolution of the Asian poultry industry and how it has affected the Newcastle disease control. I would divide the Asian poultry industry in 2 parts. I would say that there was an Asian poultry industry before avian influenza crisis back in 2004 and after the avian influenza crisis. So before the avian influenza, most of the farms were small. The biosecurity was not really a proper biosecurity, was well done, or even non-existent. And you have some illustrated pictures to understand what I'm talking about. But after the crisis in 2000, that started in 2004, the industry was forced to change because the avian influenza was very much present in countries like Indonesia, countries like China, Vietnam, and eventually some outbreaks in Malaysia or some other countries like Bangladesh. the industry had to change. And today we see much modern houses, big farms, much better biosecurity. So let me walk you through what has happened to the Asian poultry industry after 2004. The first point is that the Asian poultry industry is very much developed in this part of the world today. You can see by this map produced by FAO, and this map shows the distribution of poultry production across the world. And you can see here in Asia, these red spots, this is where the chickens are located. You see the amount of birds in this part of the world. So if there is this amount of birds, the possibility of spread of disease it's very huge, right? It's very big. So this is one point that has happened to poultry production here. It's grown exponentially. So we have chickens everywhere. But not only this, as I mentioned, the farms have become bigger. Today, instead of several small farms, the companies are investing in this type of big farms. And this is understandable because you build a big farm, then you have proper biosecurity. So you reduce the risks of the exposure to avian influenza or Newcastle disease. The drawback of this new setup is that if there is an infection with Newcastle disease virus or an infection with avian influenza virus, for instance, the losses, the economic losses are huge, right? But this is one reality here in Asia now, big farms. And this is also large production areas. These are two aerial pictures. I got them using Google Earth. And you see two producing areas in Indonesia. One in Java Island, on the left-hand side, and another one in Sumatra Island. But you see the amount of houses, the amount of layer houses, belonging to different companies, belonging to different owners. So you imagine how difficult it is to have proper biosecurity in a situation like this. But this is what we have to deal here in Asia. Okay, there is another point that is happening here today. The farms are moving from open-sided houses towards environmentally controlled houses. And today, even broiler cages are growing in some countries in Asia. So what does that mean? It means that the number of birds per square meter, or if you prefer, the number of kilograms per square meter has increased. In the open houses was probably 25 kilos per square meter. Today, they can grow 42 kilos per square meter. So there are much more birds per square meter. If there is a disease, you can imagine that the losses will be extremely heavy to producers. Another one, and this is probably one of the major threats or the major challenge for producers. You can have a very nice farm, and this is illustrated by this one in the middle of the screen, but this farm is surrounded or may be surrounded by farms with not so good biosecurity. And there is a constant threat for introduction of pathogens. It starts with avian influenza, but it can be Newcastle disease. So even these big farms with high level biosecurity, there is this threat of constant introduction of pathogens. So this is something that producers are always concerned about, right? But let's start talking a little bit more about Newcastle disease. The first point that you have to understand is that Newcastle disease is very much present around the equator. And this is in this area. It passes in the Southeast Asia, in the northern part of Africa, in the Latin America. This area, usually the countries are enzootic, or the disease is enzootic. The virus is present, is always challenging the birds, the birds and of course the producers. If you go up north, like USA or Eastern Europe or Western Europe, the likelihood of Newcastle decreases. And the same thing when you go toward the south of the equator, like in Brazil or Argentina or Australia, the likelihood of having Newcastle disease is decreased. However, you remember the industry is growing in this part of the world. The industry is not growing in Europe. The industry is not growing in the United States. The industry is growing in India. The industry is growing in Indonesia. The industry is growing in Mexico. And all these areas are full of Newcastle disease virus. Or we have to do something to reduce the losses, or to do something to be able to grow chickens in an area that is very much affected by Newcastle disease virus. So to make a short summary, this is where the virus are most prevalent. In Asia is really the genotype 7. The exception is India and Pakistan, that the most commonly circulating virus are genotype 13. But even this genotype 13, they were previously classified by DL, in this group back in 2012. This genotype 13 was previously classified as genotype 7. So in Asia, we could say that very much, very similar to what happened in the Middle East, is the genotype 7 virus that is really threatening producers here in this part of the world, right? Well, but after saying all of this, what would be the main strategies against Newcastle disease here in Asia? And the strategies have to be very well defined, because as you understood, is enzootic in this part of the world. There are many farms. These farms are extremely big. They are able to hold more than 1 million birds at any time. So you see that it's very challenging to keep the disease outside of the farms. So there are 3 main strategies. The first one is biosecurity. So when the companies opted to build those large farms, they knew that they were putting all the eggs in one basket. They knew that they would have many birds in the same area. So if they had any disease, the cost or the economic losses would be huge. So they invested a lot in terms of biosecurity. One example, they started building those farms in a more distant ways, not really close to other farms. So they wanted to have those farms separated from the traditional areas of production. Another example, several farms, even broiler farms. Even broiler farms, you have to take shower and to change the clothes before entering the farms. So this is another way to explain the biosecurity in those farms. So in one word, the biosecurity from 2004 onwards, the biosecurity has improved considerably. But not only this, the second point was vaccination. And the vaccination means producers want to have clinical protection. Yes, that's very important, but they also want to have containment of the virus spread. If the biosecurity, for any reason, it does not work, and the virus can enter in that farm, the vaccine has to give clinical protection, but has to be able to contain, to minimize the spread of that virus in that population, or even to the neighboring farms. So vaccination has to address these 2 points. And finally, and very important, is a proper monitoring of the situation. So to have a baseline of vaccination, have a close follow-up of the flocks using serology or using molecular tools, but you have to know what is going on. on those farms, right? So basically, this is what has been done here in Asia. It's not different. I bet that this is the same approach that is being done in Saudi Arabia or any other country. Okay, here there is a heavy investment in biosecurity, a heavy investment in vaccination, and also a very close monitoring to follow up everything. Well, let me go a little bit in terms of vaccination. We don't have time to explore deeper these three different points, but vaccination against Newcastle disease. What are the major drawbacks that producers face here in Asia, and what type of technology today they are using more to protect against Newcastle disease? The first one, since this disease was first described in 1926, so it means 95 years ago, almost 1 century ago. So since this disease has been described, several types of vaccines have been developed. The first ones, they tried to do attenuation of the virulent NEV. They were not very successful. They tried to produce killed vaccine. Also, they were not very successful. But this was the first attempt in terms of vaccination in the 30s. And then in the 40s, there were development of several mesogenic vaccine strains, Roquin or Quetzal from India. So several mesogenic strains were developed in the 40s, but also two well-known vaccines, Lentogenic vaccines, HB1, Kitchener-B1, and Lesotho strains. They were developed towards the end of the 40s. So for many years, nothing happened in terms of new vaccines. In the 70s, there was a kind of boom of researchers looking for thermostable strains. In those days, what they wanted was to develop a vaccine that could be given through the feed. So this would help small producers, smallholders, because they could spray this vaccine in the feed, the chickens would eat, and then they would be immunized. So several researchers worked on this, but it never came to be a success. In the 80s, some of you might remember, it was the time to develop clone ND vaccines. And one company really developed one very successful vaccine in Clone 30. They were looking for vaccines that were protective, but not as much as reactive as the old strains like mesogenic strains or even the Lesotho. So this was the first movement towards safer vaccines. In the 90s, the apoptogenic enteric strains were introduced. These vaccines are very, very, very much used till today. They do not replicate massively in the respiratory tract. They replicate in the gut, therefore preserving this respiratory tract. So these vaccines are extremely safe and they are used to date. In 1994, to be exact, the first vector vaccine was launched in the market. So exactly 26 years ago, the first vector vaccine was introduced. That vector vaccine was an FP-based Newcastle disease vaccine. This vaccine used the smallpox virus to express the hemagglutinin and also the F protein. This was the first vaccine introduced in the poultry market back in 1994. And more recently, in the early 2000s, two types of vaccines were introduced. One was a genotype matching vaccines, and the other one was the vector HVT ND vaccines. So at the end of the day, there are several options of vaccines, several types of vaccines. I just put some of this picture illustrates, I don't know, a few dozens of products, but I can tell you, there are hundreds of Newcastle vaccines out in the market. And we have to always to ask ourselves, if we're dealing with a virus, Newcastle disease virus that contains only one serotype, and we have vaccines, live vaccines, killed vaccines, why are we still here discussing about this virus? This is not like a coronavirus, like infectious bronchitis or like avian influenza. It's only one serotype. Why there's still challenging producers? So there are some reasons for that. Okay, when we are dealing with conventional vaccines, and conventional, I mean here, live and killed vaccines, there is one problem that that producers would face, and this is interference with maternal derived antibodies. Let me show you one work done several years ago, back in 1978, by a group of French researchers, Benajant. And these guys, they did a very interesting trial. Look at this. They vaccinated chickens with or without maternal derived antibodies. So they took those birds and they vaccinated them by eye drop, one dose, with HB1, HB1 strain, and they had several birds vaccinated. So from time to time, they went to that particular room, and they took some birds and they brought them to an isolator, and they challenged those birds using a Lentogenic Newcastle Disease virus. And they measured the protection. Look what they found. When they vaccinated the SPF birds, or birds without maternal derived antibodies, without passive immunity against Newcastle disease virus, this was the protection. When they challenged at 0 days, they found 60% of protection. When the challenge was done at 5 days, they got 80% of protection. At 10 days, they got almost 90%. So all the way to 60 days, you see very high level of protection. So this is what the vaccine is able to do in terms of protection. But interestingly, when they took birds with maternal derived antibodies and they vaccinated the same vaccine strain, they vaccinated the same dose using the same route, and they challenged with the same virus, this was the protection that they were able to get. In the beginning, you see in the 0 days is only 4 hours after vaccination, they've got even better protection, it was 90%. At 5 days, it reached up to 100%. But this is because of association of the local immunity induced by the live vaccine plus the maternal derived antibodies. But at the end of the day, when the challenges were done at 10, 15, 20, all the way to 60 days of age, there was a gap in protection. And this is explained by the neutralization or interference, if you prefer, between the vaccine and the maternal derived antibodies against Newcastle disease virus. And the bad news to Asian producers is that the higher is this level of maternal derived antibodies, the wider, the bigger will be this gap between the protection that the vaccine should induce and the protection that the vaccine would actually induce. The higher the level of maternal derived antibodies, the bigger is the interference, right? And this is the research did it with live virus, live in the vaccine, but they repeat this with killed vaccine, the same methodology, but the birds just received the killed vaccine. And you can see exactly the same picture, right? You see a gap between the protection in SPF birds or birds without maternal derived antibodies and birds with passive immunity. And again, the bad news for Asian producers is that Newcastle disease is a challenge. This is an ever-present challenge. So these producers, do vaccinate very heavily the breeders. So when they vaccinate these breeders very heavily, they are going to have a transfer of passive immunity to these broilers. And therefore, you're going to see this interference that you can see in your screen right now, right? So this is was presented by Dr. Ayatullah Natsir from Indonesia. He measured the level of passive immunity in Indonesia. Each bar, you can see here, each bar represents one flock. He took samples from 215 to 120 flocks, and the average of all of them was 8 log 2. Imagine, 8 log 2, there was the maternal derived antibodies against Newcastle disease. So in a situation like this, it's extremely difficult for a vaccine, a conventional vaccine, to take. So this is what we have to face here in Asia. We have lots of birds, lots of farms, an ever-present challenge. And when we place these chicks in the farms, they carry a huge amount of passive immunity. They have high levels of maternal derived antibodies. This is the challenge that we have to face. So what is the trend here in Asia? And it's not a trend anymore. Probably today is the most used vaccine in this part of the world is the vector HPT NDV vaccines. Let me take this opportunity to show you a little bit about this technology. Let's imagine that you have a Newcastle disease virus. So the most important part of this virus to induce an infection, to be able to infect the cells, is this F protein. So it's the same thing that we are hearing today on TV when you talk about coronavirus, COVID-19, that the most important protein of the coronavirus is the S or spike protein. For the Newcastle disease, the most important by far is the F protein. So what the researchers did, they took one virus that was the HVT, they detected the insertion virus, that blue line in the DNA of the HVT, and they inserted the genes of the Newcastle disease virus that would give origin, that would produce the F protein. So by doing that, when this HVT replicates, the HVT will induce the protection against Marex. You see that the antigens to produce the protection against Marex are still there. But this HVT virus will also express the F protein that ultimately will trigger the immune system, and the immune system will produce the immune response to protect against both HVT, but also Newcastle disease virus. So in a very short, in a very summarized way, this is what a vector HVT-based ND vaccine is, or we call it vector HVT ND, right? And one of the major advantages of this technology is that it's able to bypass that wall of maternal-derived antibodies. You remember the graphic from Indonesia? For conventional vaccines, it's like a wall. The conventional vaccine comes, but is not able to pass that wall. When you have this type of technology, there is no Newcastle disease virus. So this virus is able to pass through that wall. It's not neutralized by the maternal-derived antibodies. It's not neutralized by the passive immunity, right? So this is one of the major advantages of this technology. And let me illustrate this. As I was mentioning, Indonesia, this was one trial that was done to compare different technologies. The first one, commercial broilers with maternal-derived antibodies of 7.4 log 2, I apologize myself, 7.4 log 2, very similar, you remember, to the 8 log2 that was identified in the frogs in Indonesia. So very high level of maternal-derived antibodies. And these birds were split in 5 different groups. One group was vaccinated with the genotype 7 killed vaccine, or genotype matching vaccine, if you prefer. Another group, and also a live and DIB vaccine. Okay, the other group received the genotype 2 concentrated killed ND vaccine, and live and DIB. The third group received a vector HPT NDV plus live and DIB. Another group, only the live and DIB, and one group was not vaccinated to see whether that challenge was strong enough. So, and another point that was very interesting, because the challenge was done at 25 days of age, very early. But why 25? Because in Indonesia, they like small birds. So the age of slaughter, the age that they sell birds is around 28 days, 30 days max. It's very similar, as far as I understand, it's very similar to your situation in Saudi Arabia. The birds are small and they are slaughtered in a very young age. So this should be very similar to what you face in your own country. And during this, the 14 days, the clinical protection was assessed, right? So, and you can see very quickly here, the difference in terms of technology. The 2 killed vaccines, they gave between 25 to 30% of protection, very similar to that green bar that was live in the IB. But when you have the vector HPT and the V vaccine, the protection was much, much better. Just because, as I explained before, this type of technology is able to bypass, is able to go through that wall of maternal-derived antibodies. And it replicates, it expresses the F protein, and the F protein will trigger the immune system, culminating with protection against challenge. So this was really one of the reasons, probably the major reasons for a quick adoption of this type of technology by Asian producers. But when you say vector HPT and DV vaccines, we tend to think that all of them, and there are several commercial products today, we tend to believe that all of them are the same. They are vector HPT and DV vaccines. But actually, they are not the same. And let me explain here. I just choose four different products that are being commercialized out there. The first one, they use the FC126 strain. And you can see UL, it means unique long, is one part of the DNA of the HPT, and the US is unique short. So this first product, the strain that was used to remove the F protein or the genes that will produce or will encode the F protein, the F gene, is the D26, and the insertion site is between the genes 45 and 46, and the promoter is the back promoter. The second vaccine here, the second product, the insertion site is unique long as well, but the insertion site is different, the promoter is different, and even the strain from which they took the genes to insert is different. The third product is unique short, and you see several differences between the products that are available. So if these vaccines have different constructs, if they have different insertion sites, different promoters, different HPT strains, they will have different biological properties. In other words, they will have better or not so good protection, right? And here down below, I just noted all the vector products. These are commercial names that are available out there. And I can tell you, none of them are equal to each other. So vector HPT vaccines, they are different, right? I just want to illustrate, showing two different trials, comparing a vector vaccine A against a vector vaccine B. So this was published a few years ago in a congress in France. And in these comparative trials, the authors compared these two different vaccines and they challenged the birds at different timing. And you can see that the vaccine A was much, much faster than vaccine B to give protection. In this trial, the two reached very high level of protection, 95 to 100%. The vaccine A was 2 weeks before. In the second trial, again, very similar protection was achieved, again, 2 weeks before. And the last one in layers was even, this gap was even bigger, almost 3 weeks difference to reach the same level of protection. As I mentioned to you, these products, they are different. So they have different biological properties. They behave differently when they are applied to chickens, right? This is one example. The second one, I choose one comparative trial done by a university here in Thailand, very close to Malaysia. And this research group from Chulalongkorn University, they had caught female broilers and they divided in 3 groups. One was vaccinated with vector A, another one was even another product, a vector C. And they challenged these 2 products, these two groups, at 28 days of age, and they assessed several parameters. I'm going to show only one, the protection, but they assessed several different parameters. And again, you can see that the product, that vector A, was much faster, or at 28 days of age, gave 100% of protection, while the other vector gave only 78% of protection. So all of them, I reinforce this point, all of them are vector vaccines. However, they don't behave the same. They are not the same product, right? But let me move on. What this type of vaccines, and this can be any one of them. I'm going to show the result of one of these products. What you can expect from this type of vaccines under laboratory conditions. I just selected here some challenge studies with one of these vector vaccines. And you can see that there are 66 studies in these 2 different tables. And if you look at the second column, you're going to see the genotypes. Okay, and so they have studies done with genotype 2, genotype 4, genotype 5, genotype 7, genotype 8, genotype actually more than several subgroups of genotype 7. And one thing was interesting, all of them gave very high level of protection. And with genotype 7, there are 87 different challenges. And all of them, without exception, reached 95 to 100% of protection. So this type of technology really gives very strong protection, no matter what type of genotype we're dealing with. And this is important to go back to the basics. Yes, they are different genotypes. But as I mentioned before, Newcastle disease virus is only one serotype, right? So that's why you can see protection against all different genotypes. And another point that was interesting that you can see that made this type of technology so much used in this part of the world is the duration of protection. So that's why number producers or layer producers or breeder producers are using this type of technology is because of duration of protection. Let me show you one trial that was done in commercial layer pullets that were vaccinated with a vector HVT and DV product, right? And then there were other subgroups, vector plus live vaccine, vector plus live and kill vaccine. I just selected one group. I just selected the group that was vaccinated with vector HVT and DV, and then the challenge all the way to 72 weeks of age, as you can see in your screen right now, right? And then it's very interesting that these birds were vaccinated day of age by subcutaneous route, and they were never revaccinated. And look at the protection all the way to 72 weeks. Imagine that we would vaccinate some flocks today. We are in April 2021, and we would challenge this flock in the beginning of 2023 using one vaccine. And this vaccine is still able to give 100% of protection. So this changed completely the way we look at vaccines and vaccination, right? So this technology can bring all of these that I mentioned to you before, very high level of protection against all genotypes. It can bypass maternal derived antibodies, and for long-living birds, it gives a long-lasting immunity. And it was measured in this publication all the way to 72 weeks, right? But now I'm coming to one point that is very important to producers. You remember how the industry is organized here in Asia, right? And this is becoming very clear to us because of the COVID. Look at this, this illustration that was published by the New York Times. And this simulates the spread of the COVID, right? And this, in this case, is only one person passing the virus to two other persons. So the R is two. One person contaminating two others. And you see that the growth of the infection is exponentially. And this illustration shows very nicely what would happen if we are able to reduce this transmission, if we are able to break this transmission. Look how it will happen. If we're able to block, look at that first transmission, look at the amount of people that would not be exposed to this virus, right? So when we are dealing with human beings, we are able to do or to propose the social distancing. When we're dealing with human beings, we are able to propose or to wear masks, right? So to reduce the likelihood of the virus transmission, but not with chickens. Chickens, there is no way to reduce. They are going to be in the farms. They are going to be very close to each other. So there is no way that I could use this to reduce the transmission of virus in the poultry industry. What else could we do? So this is a very interesting point, and we have to always to remember what we are facing here in Asia. These are again illustrative pictures, but we have very big farms, right? In this farm, I was curious enough to count. There are 74 houses, and these houses are able to accommodate thousands, 20,000, 30,000 birds, each one of them. So there are farms today that are able to hold more than 1 million birds. In this case, 1 million broilers. And there are several farms located in the same area. This map illustrates the Delmarva Peninsula in the USA, but this is absolutely the same situation here in Asia or in any other area in the world. When you have a producing area, there are lots of farms in that specific area, right? So what would happen in this situation? This is theoretically, but let's imagine that I have one area here in Asia or in Saudi Arabia, if you want, and there are several different farms in that area. Let's take a look in this farm in the middle, the blue farm. Let's take a look at this farm. So we have a farm, several different houses, probably able to hold several hundreds of thousands of birds, right? And imagine that this farm has a good biosecurity. You have to shower in, maybe shower out. You have to change boots. You have to, but for any reason, there is a break in biosecurity, and there is a Newcastle disease virus entering this farm. So what you can expect if these flocks are not properly vaccinated, or if these flocks are not well protected, what's going to happen? The virus will replicate in those birds. They will have probably mortality, but most importantly, the amount of virus that will be re-excreted by those flocks is very, very, very high. And this virus will reach other houses. At the end of the day, this would be a disaster. All the houses probably will be affected. The mortality will be huge, and the consequences will be enormous. But not only this farm could spread the virus to neighboring farms or even to other regions. So you see the challenge that the poultry industry has today to try to reduce the transmission, to try to reduce the re-excretion of the virus. So the question that comes out is that, can this vector vaccine that I just showed to you reduce or collaborate or help to reduce the shedding of the virus or the transmission of the virus? Let me show you one first trial that was published a few years ago on the avian disease. And these authors worked with broilers, again, 7.13 log 2, so moderate to high level of maternal derived antibodies. And these birds were vaccinated only with a vector NDV vaccine by subcutaneous route at day of age. There was another group that was not vaccinated to serve as a control group, right? So, and these birds were challenged at 20 days or 3 weeks, at 27 days, 4 weeks, or at 40 days of age, okay, approximately 6 weeks of age. So with a 10 to the power 5 EID 50, with a genotype 5 strain, Chimalhuacan strain that is originally isolated from Mexico. So, and then the clinical protection of these birds, these different challenge points were measured, but most interestingly, oropharyngeal and even cloacal swabs were taken at 3 days post-challenge to measure the amount of virus that was being re-excreted by those birds, right? Look at this, the results. First, clinical protection. Well, this is what I mentioned before. The vector vaccine is able to bypass the maternal derived antibodies, log 7, log 2 in this case, and was able to replicate, to express the F protein, to trigger the immune system, and to get the protection that you see on your screen right now. So at 20 days, the protection was a little bit above 80%. At 4 weeks, almost complete, 95%, and 6 weeks, 100%. Right? This is clinical protection. This is the first point of what is expected from a vaccine. Remember, clinical protection. But then, as I mentioned, the authors measure the re-excretion of the virus. And look what they saw. So they measure the re-excretion in the oropharyngeal swabs, but also cloacal swabs to see the re-excretion by the respiratory tract, but also by the feces, right? And look at this. When they did the challenge at 20 days of age, there was a reduction approximately, given or taken, 2 log 10. So approximately the birds that were vaccinated, they re-excreted approximately 100 times less virus to the environment as compared to the control group, right? But this vaccine remains replicating in the birds. The vector vaccine, the HVT, remains replicating and remains expressing the F protein. The immunity continues to grow up. And look now when they did the challenge at 27 days and they measured the re-excretion. So at challenge at 27 days, the re-excretion, the gap, the difference was much bigger. Not this 100 times anymore. Now was somewhere between 4 log 10, it's 10,000 times, to 5.7 log 10. 10,000 to 100,000 times less, right? But as I mentioned before, the HVT keeps replicating. The HVT, the vector, keeps expressing the F protein, keeps triggering the immune system, and the protection gets stronger or deeper, if you prefer, as the time goes. And when the challenge was done at 27 days of age, at 40 days of age, look at the difference. Now, the difference is almost, or 7 log 10. 7 log 10 means that there was a reduction of 10 million times. 10 million times less virus, or 10 million fold, if you prefer, less virus being re-excreted by the vaccinated fox as compared to the control group. So this type of technology offers really a sharp reduction of shedding, reduction of re-excretion. But now there is an even more important question to ask. Is this technology able to reduce transmission? Is this reduction in shedding able to reduce the transmission of this virus to other birds? Right? So there is a very recent publication done by a group of researchers. And let me explain in a different way. I'm not going to read this, but let me explain what this group of researchers did. Well, first they vaccinated birds at day 1. One group received this vector HVT NDV vaccine. So the yellow birds, yellow bagel chicks, they were vaccinated with vector HVT NDV vaccine. The second group here, the orange birds, they were the unvaccinated control, right? These two type of birds. Then the next step, they grew these birds all the way to 42 days of age. And then at 42 days, what they did, they challenged these two groups with a genotype 7.2 virus. They took those birds, the yellow birds that received the vector HVT vaccine, and the orange birds, they didn't receive the vector vaccine, and they challenged them with this genotype 7 virus. And the next step was to mix this virus with other group of virus, other group of birds, to see whether this type of vaccine would be able to reduce the transmission of the virus. So 8 hours after these birds were challenged, these yellow birds, they were commingled with the blue birds. And these blue birds, they were vaccinated with the vector MVV vaccine. And here in the bottom part of the slide, the green birds were mixed with the orange birds. So at the end of the day, this is what was done. The yellow birds that were vaccinated and challenged were mixed with the blue birds that were unvaccinated and they were not challenged. And the other group, the orange birds that were challenged, they were mixed with the unvaccinated green birds, right? And for the following 14 days, they measured the mortality, they measured the virus re-excretion, and this was something very new in this experiment. They measured the virus re-excretion oral and cloacal daily. Every day, this group of scientists measured the re-excretion of the virus to see how it would behave, right? So let's see what happened. The first one, protection. First one, survival or protection after challenge. And you can see the group that was vaccinated and challenged, the yellow birds, they had 100% of protection. And very interestingly, when these birds were challenged and put together with the blue birds that were vaccinated and unchallenged, even the blue birds didn't die. So they were completely protected. On the contrary, the unvaccinated, the orange birds and challenged, they started dying after the 5 days, and all of them died, 100% of mortality, so 0% of survival rate. And interestingly, when these birds were put in contact with non-vaccinated birds, they were able to spread the virus, and even the unvaccinated birds, the green birds, all of them died. So they were not able to control the transmission. So after that, they measured, as I mentioned to you, they measured the shedding, the oral nasal shedding, the upper part of the graphics, and the cloacal swabs, the shedding through the feces. And you can see that the vaccinated birds, the yellow ones, they re-excreted much less virus as compared to the birds, the orange birds that were not vaccinated and challenged. It was significantly, statistically significantly lower than the unvaccinated control. And this was true to the oral nasal swabs, but it was also true to the cloacal swabs. And you can see in the unvaccinated birds, unvaccinated groups, they stopped the shedding. You remember? Because all of them died. And the group that was vaccinated, they were all the way to 14 because none of them died. They were clinically protected. So there was no mortality. So we can see that the shedding was significantly reduced in vaccinated birds. Okay? But not only this. When this is measured, the birds that were commingled, you remember the blue birds and the green birds, there was also a statistically significant difference in terms of virus re-excretion. And you can see in this group, in the blue arrow, in the blue tables here, that the amount of virus is very low as compared to the unvaccinated group. So the shedding was able to control the transmission. The virus was not transmitted in high quantities to this vaccinated group. So the replication of this virus in this group was minimal, was very low. contrary to the unvaccinated birds, they ended up dying. Right? So, in conclusion of this study, first, the shed was significantly reduced, but when they calculate this, all of this is a long calculation, but at the end of the day, the reproduction ratio was calculated to be 0.82. It means that one bird would be able to infect 0.82. And the unvaccinated birds were able to spread the virus to 3.2 other birds. If you come back, if you remember the drawing or the example of the New York Times for human beings that I showed in the beginning, you remember one human being was contaminating two others. So the contamination or the spread of the disease is exponential, right? However, and this is very important, when the R is lower than 1, the infection does not go through, the infection does not continue. So the infection to continue, the R has to be more than 1. If it's less than 1, the infection cannot continue, because one human being, in the case of COVID, for instance, would not contaminate another. So at the end of the day, the infection, the transmission, the infection would fade away, right? So 2 conclusions. The first one, this type of technology is able to reduce the virus re-excretion, as it was shown in the first trial, reducing up to 10 million times, but also this type of technology is able to reduce significantly the transmission of this virus. And this is exactly what producers here in this part of the world want. They know that the challenge is, it's likely to happen. We live here in Asia in an area where the Newcastle disease virus is endemic. If there is any problem with biosecurity, this virus would infect Bangkok. This might happen. So what producers want is really, if it infects that specific house, the amount of virus that would be re-excreted to other houses is very much limited by the vaccine. But the other houses would have been vaccinated with also that vector HVT and EV vaccine. So I would need much more virus to have infection. So at the end of the day, the infection would stop. The infection would not progress. The infection would not reach all the houses, or even more important, the virus would be released or would be getting out of this farm in a very, very limited way, or in a good way, it would even not happen. So the virus would be contained. The outbreak would be contained in that specific farm, not going out, not contaminating, not infecting neighboring farms, or not infecting neighboring regions. So this is what producers here in Asia are using very much now. Right? I'm coming to the end of my presentation. It's 1 hour. So I would like to finish with 3 different methods. The first one is that Newcastle is an ever-present problem or ever-present challenge here in Asia, right? Sometimes it goes up, sometimes you feel that the disease is not a challenge anymore, but it comes back and goes up, it comes back. So it goes and it comes back, goes and comes back like a wave. So this is a normal behavior of this disease in a given region or in a given country. But the virus never goes away. So this is important, right? So, and despite being reported almost 100 years ago, remember, 1926, Newcastle disease remains as one of the major threats to poultry producers here in Asia, right? Here in this part of the world, with the development of the industry, remember, bigger farms, more birds in the same area, populational density very, very big in the producing areas. So having a proper prevention against this disease is critical to keep the profitability of those companies. Without this, those companies could not survive. So I could say that these companies have invested heavily on biosecurity, have invested in proper vaccination, and most of them today are adopting this new technology that I mentioned to you. And another very important point, they have a very close monitoring of the situation. So these are the three cornerstones, the three most important pillars if you want to prevent, if you want to reduce the losses against Newcastle, caused by Newcastle. Apologize. Okay, so this is what I had prepared for you. I hope you could get a glimpse of what we have done here in Asia, what producers are doing here in Asia to reduce the losses related to this disease. Thank you very much.
Thank you, Dr. Marcelo, for your nice big TV presentation and for sharing your experience from the Southeast Asian countries with us. And I will make a short Arabic summary for your presentation, then we can start the Q&A session. احب بس اذكر حضراتكم لو في اي حد ليه اي استفسار يقدر يكتبه في الاسئله والاجوبه في البلوك بتاع الاسئله والاجوبه، وهنجاوب عليها في نهايه المحاضره. في البدايه دكتور مارسيلو استعرض معانا المرض في نيوكاسل وانتشاريته من سنه 1922 ل 29 الى الان. هو مرض منتشر في كل مكان، تقريبا 16 دوله بس حوالين العالم اللي ما سجلوش المرض، وهم عباره عن جزر صغيره في المحيط الهادي. اهميه المرض ان هو بيشكل خطوره على الامن الغذائي. واستعرض معانا ان في دول شرق اسيا الصناعه منتشره وبكثافه وبيعتمدوا على دواجن بشكل كثيف، لكن في فتره زمنيه ما بين ما قبل 2004 وما بعد 2004 ان الصناعه ما قبل 2004 كانت مزارع صغيره الامن الحيوي فيها غير جيد. ما بعد 2004 وليه 2004 بالتحديد اللي هي انفلونزا الطيور، الصناعه بقت اكثر تنظيما، مزارع كبيره بتهتم بالامن الحيوي بشكل كبير. واستعرض معانا انتشاريه المرض تقريبا منتشر في كل دول العالم، لكن الدول خط المنتصف اللي هي الحراره فيها عاليه بينتشر فيها المرض بشكل اكبر، وبيكون اقل انتشارا كل ما طلعنا ناحيه اوروبا وامريكا او حتى نزلنا ناحيه الدول الجنوبيه. طبعا كان تأثير التحول من المزارع الصغيرة للمزارع الكبيرة بشكل كبير على الكثافة بتاعة الطيور، لأن في المزارع المفتوحة كان بيربوا تقريبا 22 كيلو على المتر مربع، في المزارع المغلقة والمحكمة بيتم التربية في حوالي 33 ل 34 ل 43 كيلو جرام على المتر الواحد. فده بيسبب لنا مخاطر اكتر في حاله حدوث اي امراض او انتشار اي وباء. وبعدين استعرض معانا انتشاريه المعزوله اللي هي الجينوتايب 7 وقد ايه هي انتشرت في نهايات التسعينات من بدايه نهايه الالفينات من دول شرق اسيا للشرق الاوسط وحتى لامريكا اللاتينيه. واستعرض معانا ان الاستراتيجيات اللي بيعتمدوا عليها في دول شرق اسيا بتعتمد على 3 محاور رئيسيه: الامن الحيوي، المنع، والتحصين عن طريق التحصين للحمايه الظاهريه والحمايه للامراض للمرض والاعراض والنفوق، وكمان منع انتشار الفيروس في حاله حدوث الاصابه. واخر حاجه المونيتورنج او بنسميه المتابعه الحقليه للطيور للاصابات والمناعات، سواء بالطرق السيرولوجيه او بالطرق المولكيولر اللي هي الجينيه. نقطة مهمة استعرض معانا من ساعة ظهور المرض سنة 1926 إلى الألفينات إلى يومنا هذا، إيه هي التطور بتاع اللقاحات، وإن هو ابتدى بأول دراسات في بداية في الثلاثينيات، وبعدين ظهور أول لقاحات من متوسطة الضراوة إلى خفيفة الضراوة زي الكوماروف أو الهيتشنر واللاسوتا في الأربعينات. وبعدين في السبعينات كان في بعض الدراسات ان هم يعملوا لقاحات تبقى ثابته حراريا بحيث يقدروا يضيفوها على العلف، لكن التجارب دي ما نجحتش. في نهايه الثمانينات بدات تظهر الكلون فاكسين، اللقاحات المستنسخه، بحيث نقدر ناخد حمايه جيده كمان برد فعل اقل من اللقاحات اللينتوجينيك العاديه. بعد كده في التسعينات بدأت تظهر العترات المعوية الغير ممرضة، اللي هي بتبقى تقريبا تدينا حماية لكن من غير أي أعراض جانبية للمرض. وفي بداية الألفينات بدأت تظهر اللقاحات المحملة، وكانت أول تجربة للقاحات المحملة بتحميل النيوكاسل على جدري الطيور. وبعدين بدأت تظهر اللقاحات المحملة على مرك في الألفينات، سنة 2000 وما بعد الألفينات، نهاية 2010 وهكذا. موجود مئات من لقاحات النيوكاسل من شركات عديدة، ولكن ما زال موجود المرض. طيب ايه هي المعوقات اللي كانت بتبقى بتقابلنا في المرض وانتشاريته ليه موجود لغاية دلوقتي؟ استعرض معانا دراسة مهمة بتقول أول نقطة كانت الأمان الحيوي مع المناعات الأمية، عذرا، وجود حاجز مناعات أمية عالي. واستعرض معانا دراسة بتبين التأثير بتاع المناعات الأمية دي في حالة وجود التحصين سواء حي أو ميت، من هو عمل اختبار تحدي لطيور ما فيهاش أي مناعة أمية ومحصنة، وطيور فيها مناعات حاجز مناعات أمية عالي ومحصنة، وقد إيه كان في فرق كبير ما بين الطيور اللي ما فيهاش مناعات أمية واللي فيها مناعات أمية، سواء اللقاح كان لقاح حي، ونفس الفكرة كانت مع اللقاحات الزيتية. وقد إيه هم عندهم حاجز مناعات أمية عالي، وأعتقد نفس الوضع عندنا في دول الشرق الأوسط، لأن الأمهات بيتم تحصينها مرتين وثلاثة وأربعة، فبالتبعية بينتج حاجز مناعات أمية عالي في الطيور عمر يوم. بالتبعية عمل بعد كده دراسة اختبار تحدي لطيور محصنة بلقاح زيتي من العترة الجينية رقم 7، طيور محصنة بلقاح زيتي من العترة الجينية رقم 5، أو طيور محصنة باللقاح التكنولوجي المحملة recombinant فاكسين على ماريكس. وقد ايه كان في فروقات واضحه للحمايه للاعراض والنفوق على عمر 25 يوم، ان اللقاح الفيكتور المحمل على مارك مع تحصينه حيه كان مدي فرق كبير جدا في الحمايه، تقريبا 85-90%، على عكس اللقاحات الزيتيه سواء من العترة الجينية رقم 7 او من العترة الجينية رقم 5 مع تحصينه واحده حي كانت مديانا حمايه تقريبا 30%. وبعدين استعرض معانا طيب احنا دلوقتي في لقاحات التكنولوجي بتاع اللقاحات المحمله موجوده وبتنمو من سنه للتانيه بشكل كبير. طيب في فروقات ما بين اللقاحات المحمله ولا لا؟ عندنا اكتر من لقاح محمل دلوقتي موجودين في السوق، تقدر حضراتكم تقدروا تحصلوا عليهم. فاستعرض معانا الفرق في تركيب بعض اللقاحات المحمله، هل في فروقات في التركيب بتاعه اللقاحات المحمله دي ولا لا؟ استعرض معانا بعض النقاط، ان في انواع مختلفه من ال HVT، فده فرق مكان الزرع اللي بيزرع عليه الجين بتاع النيوكاسل، نوع الجين بتاع النيوكاسل، المعزوله بتاعه النيوكاسل اللي واخد منها الجين، وحاجه بنسميها البروموتور او الماده المحفزه اللي بتتحط قبل الجين اللي انا بزرعه. كل دي فروقات موجوده في التركيب ما بين اللقاحات المحمله المتواجده في السوق حقليا، بينعكس عليها بعد كده الفروقات في الحمايه الناتجه عن هذه اللقاحات المحمله. ويستعرض برضو بعض الدراسات اختبار التحدي، هل اللقاح المحمل ده ليه تأثير إيجابي بشكل كبير على الحمايه ضد النيوكاسل بمعزولاته المختلفه، وبين لنا احد اللقاحات المحمله دي ودراسات مختلفه تقريبا اكثر من 140 اختبار تحدي، منهم تقريبا 87 اختبار تحدي للمعزوله الجينيه رقم 7 بنسبه حمايه تتجاوز 95% تصل ل 100% في احيان كتير. طيب دي نسبه الحمايه. طب مده الحمايه لو انا بحصن طيور امهات او بياض، استعرض معانا نتيجه برضو اختبار تحدي تقريبا شبه اسبوعي من عمر 3 اسابيع حتى 72 اسبوع، وبناخد الحمايه تقريبا بدايه من عمر 3 ل 4 اسابيع 100%، وبتستمر معايا حتى 72 اسبوع. النقطه المهمه والاهم في البرزنتيشن اللي هو عرضها لنا اللي هي قدرة اللقاحات دي على تقليل انتشارية المرض في حالة إصابة، في حالة الحدوث. إيه هي قدرته؟ ووضح لنا مثال من مجلة النيويورك تايمز نشرته في حالة مرض الكورونا هنا في الوقت الأخير. لو شخص واحد مصاب بكورونا عدا شخصين، الشكل بتاع الانتشارية بتاع المرض ده هيبقى شكله عامل إزاي؟ وإذا قللنا كان عندنا القدره ان احنا نقلل الانتشاريه دي من شخصين لشخص واحد، التاثير ده بيكون شكله عامل ازاي. وعكس ده على صناعه الدواجن، قد ايه هي بقت كثيفه، ووجود حظاير بشكل كبير متجاوره، حجم المزارع بقى بشكل اكبر، وجود مشاريع متقاربه من بعضها، وتاثير التقليل اعاده انتشار المرض ايجابيا على الموضوع ده. حتى في بعض الاوقات عندهم مزارع بتصل حجمها الى 74 حظيره في المكان الواحد. طيب اهميه منع انتشار الفيروس، ان انا مش هقدر امنع ان الفيروس يجي لي من الجو، لكن اقدر امنع اقلل انتشاره. طبعا اول نقطه قال لنا الامن الحيوي دي بنكرر عليها في كل محاضره، ولكن بعد كده الحمايه الناتجه جوه الطيور من اللقاحات اللي هو بتاخدها على اعاده تقليل وانتشاريه المرض ده. استعرض معانا دراستين. الدراسه الاولانيه بطيور فيها حاجز مناعات اميه عالي تقريبا 7.13 لوج 2، تم تحصينها باللقاح الفيكتور المحمل عمر يوم تحت الجلد، وعمل اختبار تحدي على 3 اعمار مختلفه، على عمر 20 يوم، عمر 27 يوم، وعمر 40 يوم، وعمل اختبار تحدي بالعتره الجينيه رقم 5، وبدا يشوف الحمايه للاعراض والنفوق لمده 14 يوم بعد التشالنج. كمان بدا يقيس كثافه العتره بتاعه اختبار التحدي كل 3 ايام باختبار البي سي ار. وكانت الحمايه للاعراض والنفوق تقريبا من عمر 20 يوم كانت 80% حمايه، الطيور الغير محصنه تقريبا كلها ماتت. العمر 27 يوم ادانا 95% حمايه، و40 يوم كانت 100% حمايه من اللقاح المحمل لوحده. طيب القدره بقى على منع انتشاريه او اعاده اخراج الفيروس تاني للبيئه، كان على عمر 20 يوم الطيور المحصنه باللقاح المحمل كانت عندها القدره ان هي تقلل اعاده اخراج الفيروس تقريبا 2 لوج، يعني تقريبا 100 مره اقل من الطيور الغير محصنه. طيب على عمر 27 يوم، نظرا لان اللي كلنا عارفين لقاح الفيكتور بيفضل يستمر في التكاثر داخل جسم الطائر مدى عمره، ويتكاثر ويطلع يخرج لي مناعه مستمره. على عمر 27 يوم كانت اعاده اخراج الفيروس تقريبا من 4 الى 6 لوج. طب على عمر 40 يوم وصل معايا منع اعاده اخراج الفيروس او تقليل اعاده اخراج الفيروس للبيئه تقريبا 7 لوج، يعني انا بتكلم في 100 مليون 10 مليون ضعف. فده بيبين لنا اهميه التحصينات في منع اعاده اخراج الفيروس للبيئه تاني في حاله حدوث اصابه. فده دور مهم للقاحات في الامان الحيوي للطيور، مش بس هتحمي الحظاير الاخرى داخل نفس الحظيره، لكن كمان هتحمي المزارع والحظاير المجاوره. في بحث حديث تم نشره في 2020 مجموعه من الباحثين على كمان النقطه دي بالتحديد. انا لو عندي طيور حصل لها اصابه وهي محصنه، وخلطتهم مع طيور محصنين بما يسميهم كانوا في احتكاك ما بينهم، عشان اشوف النقطه اللي ذكرها في حاله الكورونا، قدرة الطائر المحصن على إن هو يعدي كام طائر تاني معايا، وفي حالة الطائر الغير محصن وحصل له إصابة قدرته هتبقى تعدي كام طائر تاني في حالة المخالطة. وكان في فرق واضح وكبير ما بين الطيور المحصنة باللقاح الفيكتور على عمر يوم من ناحية الحماية للأعراض والنفوذ، تقريبا في 100% حماية من النفوذ. في حالة الطيور الغير محصنة كان الحماية للنفوذ والأعراض قليلة جدا، مع مرور الوقت تقريبا كل الطيور ماتت. لكن النقطة اللي كانت مهمة بالنسبة لنا إن أنا أقيس قدرة الطائر المحصن والغير محصن على إن هو ينشر لي المرض. كان الطائر المحصن قدرته إن هو يعدي 0.82، يعني 8 من 10 من الطائر. انما الطيور المحصنه الغير محصنه كانت قدرتها ان هي 3.2 يعني تقدر تعدي 3 طيور اخرين. يعني الطائر المحصن عنده قدره ان هو يعدي اقل من طائر واحد 8 من 10، لكن الغير محصن كان عنده قدره ان هو يعدي 3.2 يعني 3 طيور واكثر. وده لما هنطبق نفس المثال على المثال اللي ذكره بتاع النيويورك تايمز يبين لك الفرق قد ايه ما بين الحمايه في الطيور المحصنه بالفيكتور او الغير محصنه، وانعكاسيته بقى على انتشاريه المرض من حظيره للثانيه ومن مزرعه للثانيه في حاله حدوث الاصابه. فدي نقطه مهمه جدا بينظر ليها في تقييم البرنامج التحصيني او اللقاح اللي انا بستخدمه للحمايه ضد اي مرض. وهو لخص لنا المحاضره في الاخر ان مرض النيوكاسل موجود من 1926 وهو مستمر معانا الى الان. بتحصل هجمات وتبتدي تقل وتحصل تاني، وفي الغالب بترجع كمان للتراخي في عوامل الامان الحيوي. مهم بالنسبه لنا ان انا ابقى عامل برنامج تحصيني بشكل جيد يتغلب لي على العوامل اللي احنا ذكرناها، سواء حاجز مناعات اميه عالي، سواء ان انا اقدر اطبقه بشكل جيد في آسة او وما شابه ذلك. وفي النهايه يقدر يديني انواع الحمايه المختلفه، سواء حمايه موضعيه او حمايه دمويه وحمايه خلويه. فبالتالي في النهايه يقدر يقلل لي اعاده اخراج الفيروس للبيئه مره ثانيه. في النهايه امل ان انا اكون عملت شورت سمري سريع غطى كل النقط اللي هو ذكرها. I would like to invite the Dr. Marcelo to start the Q&A session. My colleague Dr. Ahmed will ask question and I will help him.
السلام عليكم ورحمة الله وبركاته. أهلا بكم جميعا. The question for you, Dr. Marcelo. The first question is, if there is any rule for vector vaccines in mucosal immunity?
That if understood well, Ahmed, is if vector vaccines would induce mucosal immunity.
Yes.
In your mic, Marcelo.
It's not. You're not hearing? Can you hear me now?
Yes, it's clear for me.
Okay, probably the battery yes, was
thank you.
Gone bad. Well, and yes, the vector vaccines, they have done some studies to understand what type of immunities this technology would induce. There are some publications about that. And this type of technology, this type of vaccine induces the 3 types of immunities. Cell-mediated, very strongly. Humoral immunity, circulating antibodies, but also they measure mucosal immunity. So it's able to induce the 3 different types of immunity that's necessary to fight against Newcastle disease.
Okay. The second question, in all studies present in broilers.
I cannot hear you.
Is it clear now? Can you hear me now?
No, Ahmed, I cannot hear you. Let me.
Can you hear me?
Marcelo, I think it's a problem in your connection.
Something is wrong. I cannot hear you guys.
Can you hear me, Marcelo? Do you hear me?
Let me try again.
We hear you quite well.
Let's try again.
Okay, can you hear me now?
I cannot hear you.
Okay.
Can you use instead of the mic, now?
Remove the plug.
Yes, do you hear us now?
Can you hear me, Hassan?
Yes, I hear you quite well.
Okay, but I cannot hear.
So you can rejoin.
Let me call somebody.
Okay.
Hey,
على هذا
Lee, please. I'm trying to fix problems related to.
Okay, no problem.
Yes, they cannot hear. I cannot hear them. They are asking questions. I cannot. No, not here. Please say something.
Marcelo.
Oh, now I can hear you.
Okay, perfect.
Okay.
Okay, let's begin again.
Thank you. We apologize for this problem.
No. The second question is, in all studies present in broilers, all focusing on shedding and protection. But what about serological evaluation in short-lived birds? And what do you think? Yes, and what do you think about the killed vaccine at day old with the present high maternal derived antibodies?
Yeah, well, in that trial, they evaluated the seroconversion. So in the link, you're going to see the references, you can see. But with this type of vaccines in broilers and using HI, some labs can get 5 log 2, 6 log 2 only with the vaccine. Okay. Related to the killed vaccine in broilers, actually it helps when you are using only live vaccines and you add up a killed vaccine, it helps. But we have to consider that this will be partially neutralized. And the problem is that the higher is the level of maternally derived antibodies, the biggest will be this interference. And in Asia, in Southeast Asia, where the breeders are heavily vaccinated or even exposed to the field challenge, the field virus, the level of maternal derived antibodies, the level of passive immunity is very high. So you have to consider that part of this vaccine will be neutralized. And probably this is one of the reasons why producers are really moving from killed to vector vaccines.
Okay. So, Dr. Marcelo's third question, under field conditions, we are experiencing Newcastle disease challenges before 20 days of age. Are booster doses required for such condition? Which age will be? And if yes?
Okay, then first, thank you for this question. It's an excellent question. As you understood, the vector vaccine is the F gene is inserted into a Marek's disease virus, an HDT. And the Marek's disease virus replicates in cycles. So the protection will grow as long as the virus keeps replicating. So in the beginning of the life, the protection induced by this vaccine will be relatively low. So at 2 weeks, we can expect, let's say, 20, 50% of protection at the most. At 3 weeks, we are going to have 89%. At 4 weeks, complete protection, right? So, but I have to protect my flocks, as in this question, The fox when they are younger than 20 days, when they are 14 days, when they are 16 days. And for that, we need this type of technology needs that it's a live vaccine in the hatchery by spray. And in case of very heavy challenge, we do recommend a booster at farm level at around 8 to 10 days. 10 days should be okay. Okay, and then we would cover these first 2 or 3 weeks, and then at the rest of the life, the vaccine would express its full potential. Thank you for who asked this question. Very good. Thank you.
Okay. The next question, when do we expect local immunity to kick in post-vaccination with vector vaccines?
For local, what we have measured, actually it was a group of scientists from Belgium. What this group of scientists have measured is in SPF words, is when the humoral immunity, when the circulating antibodies start to be detected. And in that specific trial that is published, this is information is available, they started to detect significantly higher seroconversion or antibody response at 9 days of age. So at 9 days, the antibody response was significantly higher as compared to non-vaccinated group. So I'm assuming that this could be the same for the local immune, that is nothing more than IgA. The cell-mediated, I would expect that would be much faster than this. Okay?
Okay.
But we, to be honest, Ahmed, we have never measured when it starts. We measure at, let's say, at 3 weeks,
yes,
we see that it's very high, or 4 weeks, it's very consistent.
Yes. So next question, what's the difference between using F insert and other vaccine using the 3 inserts, H protein, N protein, and F protein?
The difference, the insert is F and the other vaccines with 2 inserts?
Yes.
What we have seen is that first, for Newcastle disease, the F is the best one. And there are several publications dealing with that, inserting the F, inserting the hemagglutinin, or inserting the 2 of them, and measuring the protection. And the best one for Newcastle disease is the F. And that's the reason why all the commercial vaccines have the F protein. When you insert an additional insert, it might be from IBDV, or it might be from laryngotracheitis, or from any other virus. What happens is that the replication of the herpes virus of HPT will be slower as compared to the single insertion. So if, let's imagine a scenario that if I can get 100% of protection with a single vector ND vaccine, if I have a double insert, it might take 1 week or 1.5 weeks later to get the same level of protection, just because the replication of the vector will be slower. You can compare, is a truck carrying a load of some weight, and now I have the same truck carrying the double of that weight. That truck will go slower as compared to one that is carrying less weight. I hope I have explained myself.
Yes, it's clear. Using the regime Newcastle disease vector plus Newcastle live vaccine at day 1, which is better to use monitoring? HI or ELISA, target F protein, and at which age we can do this monitoring?
That's a very good question. Today, ELISA against this type of vaccines is commercially available. There are 2, to the best of my knowledge, there are 2 companies commercializing this type of ELISA. And you can use them. They are very, very much reliable. Their results match very well with HI. that somehow matches with the protection level. So you can use both of them. The most important is for the companies to establish their own baseline. So if you are one flock you measure with ELISA, the other one you measure with HI, the other one is a different ELISA from a different company, or another HI from a different sport. So it becomes very difficult to analyze. What is good is to choose one test, one lab, and then test everything according to this lab. The second point is that which age, right?
Yes.
The first one is always good to test in the beginning of the life to have an idea about your MTA. So this is not for protection purposes, but it's going to give you an information about the level of maternal derived antibodies that's being transferred from the breeders. Okay. The second information is that if I take samples at 21 days, I will be in the transition period between the passive immunity that is going down and the active immunity that is starting to going up. So at 21 days, it's not a good period because you are going to have low detection, the level, and you are not going to capture what you want. You want to capture exactly what your vaccines are inducing, right? So I would delay a bit. In Saudi Arabia, I would take at the slaughter age, I'm assuming that should be around 28 days, correct me if I'm wrong. So at that moment, you're going to be able to appreciate much more what the vaccine is inducing and not measuring the residual level of maternal derived antibodies. Okay, I would go towards the slaughter age.
Yes, okay. There is a question here, I think it's answered. Is the monitoring of Newcastle disease vaccine with ELISA and HI can be correlated? I think this part of question can be answered. Like the second part is, what is, I think, low reflex satisfying protection? I think he means that satisfied post-vaccine reaction can be handled. I think this he want to ask about.
Post-vaccination reactions, right?
Yes.
This is post-vaccination reactions is a problem. And now we are facing something that you have great experience with that. In Asia, we are facing now from 2 or 3 years, one virus called H9N2. And I know that you have vast experience with this virus. And we are learning right now because this virus has spread around the region for no more than 5 years. Yeah, I know that you have great experience with that. And one of the problems that we are learning is that when you have severe post-vaccination reactions plus H9N2, you can have, I would call it a disaster, right? One thing that it was shown recently in one trial that some of my colleagues conducted in Ukraine is that the application of the vector and the vaccine plus live vaccines, in case the trial done in Ukraine, it was 3 Lasota strains. The vector and D vaccine, HVT and D vaccine, was able to buffer the replication of the Lasota, was able to reduce the damages that the Lasota was creating in the respiratory tract. So this is another point, another good point, and thank you for this question, for the use of the vector vaccine. It does reduce the post-vaccination reactions caused by the live vaccines that you eventually apply, live and D vaccines, of course, that you eventually apply to those flocks.
I think this answer is correlated to the second question here, which is what is extra benefits of using vector Newcastle vaccine regarding other disease challenges. So you mentioned here that the first one is it's reducing the post-vaccine reaction of other live vaccines used in the vaccination program, right? And
Yes.
what else? And what else?
There could be some advantages. Ahmed. One is really to reduce the post-vaccination reactions, preserving the respiratory tract, and this is one advantage. Another thing that it was very well measured in Brazil was because it preserves this respiratory tract, it reduces the use of antibiotics. There was a publication showing that the use of vector vaccines as compared to only live vaccines was able to reduce in 40% the use of antibiotics in those lots. And this for the Brazilian industry is very important because Brazil, as you understand, is an exporting market. So to use antibiotics, to use this type of drugs, it might be a problem. So this is one of the reasons why this type of vaccines is very well accepted in that market. Although Brazil is free of Lentogenic Newcastle Disease virus. So there is this problem. There is another point that is very difficult to measure, but I'm going to mention. This is an HPV-based vaccine. Probably there is some protection against Marex as well, right? It's very difficult to measure the benefits of that if there is an impact on contamination rates or any. It's very, very difficult to measure. Or in terms of by vaccinating against Marex, you are going to avoid the immune suppression related to the field infection of that virus, of Marex disease virus. So it's very difficult to measure, but theoretically, it could have an impact because of this. Okay, these are the examples that cross my mind right now.
Okay, perfect, thank you. So, okay, what is the main disadvantages of attenuated vaccines?
Attenuated live vaccines, they are very good. We cannot say that they are good. They are good. They play their role, and they have been used since the 40s. Yeah. When Lesotho was developed by Epigenetic. So they are good. They are good. Today, with the modern poultry industry, with really too many birds in the same house, with and then you have to move away from the more reactive vaccines. You have to try to move away from Lesotho or even mesogenic strains that it's still being used in Malaysia, here where I live, or in India. So, but these strains are extremely reactive. Yes. They are extremely immunogenic, right? Extremely protective, but they are extremely reactive. Today, the modern poultry industry requests to use, safer live vaccines. But again, they are good vaccines. Don't say that they are not good. They are very good vaccines. They are very complementary to vector vaccines, as I explained before. So I don't see disadvantages. I only see that you have to use them carefully. Okay?
Perfect. Do vector vaccines give maternal antibodies in chicks when used in broiler breeders, and why?
If the vector vaccines would induce passive immunity, that's a very good question. Very good question. Most likely, some of the antibodies that are induced by the vector vaccine, by the F protein, right, will be transferred to the broilers. However, in breeders, we have so many other vaccines, live and killed vaccines, right, that at the end of the day, we are going to detect only the, let me call it, the normal maternal-derived antibodies. Okay, but most likely part of the antibodies induced by the F protein will be passed on to the progeny.
Okay, this question, I think it's answered. I would say it also again. What do you expect to happen when you have to post a Newcastle disease clone in vaccination protocol, despite presence of some uncertain marks of even influenza H9 infection?
Yes, this is what I mentioned before, and we are learning from you guys. You have more experience than us when dealing with H9 and 2. But there are several publications dealing with this issue. If you have a flock that is exposed to H9 and 2, and you have a reactive vaccine, it can be IP, it can be Newcastle disease, the reactions will be strong. So if you apply the clone 30, but if you apply them in a proper way, that you're not going to have rolling infections or excessive post-vaccination reactions, most likely you're not going to have major problems. But if you apply this vaccine, let me say poorly, reaching only 30% of the broilers, 40% of the broilers, so this vaccine will replicate in those 30, 40, or 50% of the broilers. It will jump to the other non-vaccinated flocks, so the reactions will be much stronger. And if there is H9 and 2 replicating those flocks, the results will be pretty bad.
Okay, Hazem, I think I finished here. No more questions.
There is a couple of questions in the chat. I will take them. The higher homology of the X gene from vector vaccine must match with field virus or any F gene from different genotype in terms of protection or not?
Yes, this is a very good question. Today, there is this type of genotyping the virus. So there is this belief that if I have a virus in my vaccine that matches with the virus that is circulating in the field, I would have much better protection. Today, what is almost a consensus in the literature is that there is no, you can have different genotypes, vaccine and challenge virus, and the protection will be the same. If I have a genotype matching vaccine, what I would observe is that a few times I can have less re-excretion of the virus, the challenge virus, as compared to a non-genotype matching vaccine. So, but at the end of the day, in terms of protection, it does not matter. It does not really matter if the F gene comes from a genotype 1 virus, or a genotype 2 virus, or a genotype 4 virus, or even a genotype 7 virus. There are the 4 different types of virus that are being in commercial products out there. The protection would depend much more on the speed of replication of this vaccine and the ability to induce immunity.
Okay, there is another question about the effective results of using fowl pox virus or HPT in Newcastle. Yeah, I might be he's mixed because of the vector that you mentioned at the beginning that we have pox, but it's not.
You cannot imagine how good this question is. Very interesting. Actually, let me just mention something. This first vaccine that was a vector vaccine for the poultry industry that was commercially launched in 1994 is a product called Vaccinomycin FPN. So it's commercialized by the company that I work with. And the insert is the fowlpox. So what we did probably 5 years ago, we vaccinated some birds with Vaccinomycin or the vector HPT NDV plus a vector pox ND, really to see whether there would be any difference in protection. Could be earlier protection, stronger protection. Interestingly, we didn't see any advantage of associating these two types of vaccines. But this is a very interesting question, and we did a research on that.
Is there any foul box Newcastle available in the market now, or it's not?
Well, there are, to the best of my knowledge, there is only one. It's in the USA, but it's being used only in turkeys. So it's not really very successful. And major disadvantage of the fowl is that this vaccine has also some level of interference with maternal derived antibodies. That does not happen, or happens in a much lesser extent with the HVT vector NDV vaccines.
Are there studies that have compared the Newcastle vector vaccine with the live ND vaccine regarding prevention of shedding shortly after vaccination through local immune response, T or cellular, or secretory immunoglobulin A? This might be relevant for producer in case A who slaughter birds at relatively young age and need rapid protection after vaccination.
Yeah.
He's asking about if there is studies to compare the vector versus the
live. Yes. I am not aware of any publication doing this. What I have seen is not not comparing the transmission of the virus. What I have seen is comparing the reduction of shedding. So if you want reduction of shedding, you can find some information. But I have never seen when you put these birds vaccinated with live vaccines and vaccinated with vector vaccine, and then you put them together with other birds to measure the transmission. This I have never seen.
I do believe that here's a question relevant to the amount of the shedding of the virus, because here in Saudi we are as a high risk for the Newcastle, so we are always advising the live vaccine plus the vector vaccine.
Yes. But if you want to measure only the reduction of shedding, so you can find information in the literature, not the reduction of transmission.
Yes. There is a couple of new questions. Is there any anti-vector immunity against the virus Marek used in the vector vaccine? I think he's asking about if there is a protection from the vector for the Marix itself. Yes.
Yes, it is. And this is a good point, because for each company, it's not only for the company that I work for, when you propose a vector vaccine, you have to demonstrate to authorities that the vector vaccine will protect against the target virus, indeed, in this case, but this vaccine has also to protect against Marix disease challenge. So it does protect.
We have another question which is repeated twice. How to manage the ND vaccination if you are giving a vector for layer or breeders that's long life in terms of booster dose of the live vaccine, because there is a routine vaccination with the live vaccine in our area might be monthly or bi-monthly, even in production. So what would be the change if you are using the vector vaccine, and do you guarantee that there is no production losses?
That's another good question. Guys, thank you for this question. I really appreciate that. In theory, you can reduce significantly the amount of vaccines that you are doing in the farms. Significantly. If you are able to protect the birds in that critical period between 0 and 3 weeks of age, the vector vaccine will reduce very, very high level of protection throughout the life of the birds. Throughout the life. So there are no doubts about that. Usually, what layer producers do is to have one vaccine a day of age, then they do another vaccination around 10, then 3 weeks, 6 weeks, maybe 12 weeks, one vaccine before onset of laying. And you are right. It's very common to repeat this vaccine every 4 weeks during production, every 4 weeks, 8 weeks. So what I can tell you, first, you don't need those killed vaccines that are given at mid-rearing periods. In Asia, it was very common to give an NDIBD vaccine at 7 days of age during the debeaking. You don't need. Oh, producers keep doing, but you don't need. It was common to use a killed vaccine at around 10 weeks of age. It's not necessary. However, as Newcastle disease is so threatening, if there is any break, there is the losses are so huge, producers are always a bit reluctant to remove some vaccines. And the vaccines during the production period. So in theory, it's not necessary, really not necessary. These vaccines very often are very poorly applied. They are applied by drinking water for layers in cakes. They don't reach all of them. They're poorly applied. However, when you talk to producers, it's very, very difficult to convince them to remove these vaccines. Okay, it's not. And the second question that I would like to answer: do you guarantee? Well, this is another good question. Thank you. But you know, I'm very, very much convinced about the efficacy of this technology. But guarantee is a word that is extremely difficult in our business because there are several other parameters. Immune suppression caused by other disease, right, by mycotoxins that can jeopardize the protection induced by this vaccine. So unfortunately, I want to reply to that. I cannot guarantee.
Okay, we have last two questions, one in topic and one off topic. The one in topic is there is any relation for the protection that's coming from the vector vaccine in terms of the protection for the other respiratory diseases like IB?
Okay, there is no protection, no interference, no. What is being observed in some countries, and let me mention again my home country, Brazil. In the past, they used to do a ND-IB live at day of age. With the introduction of the vector vaccine, they could remove in Brazil, that is a country low challenge, they could remove the ND live from these drugs. So the only live vaccines that are applied are the infectious bronchitis. Right, so there is no interference, there is nothing. So the protection against IB was improved, not because of the vector vaccine will improve the protection, no. But they could remove the ND live vaccine and they left the respiratory tract only to IB vaccines. And obviously, you're going to get better protection, right? So this is one advantage. But in our cases here in Asia or even in Saudi Arabia, we cannot have this luxury of removing the live vaccine, the live ND vaccine from the hatchery. So most likely we will not see much benefit in terms of protection against other respiratory diseases.
Okay, the off-topic question, and I think this is the last one. What do you expect in the future of coronavirus?
Oh, then I have thought about this a lot and about the type of vaccines that have been applied in human beings. And there are several different technologies, and I'm very curious to see. One of them is the inactivated vaccines, right? This type of technology is very safe. There are inactivated human vaccines for I don't know how many years, so I'm not really afraid of it. However, based on what we know in terms of vaccinology in poultry, if there are variant strains of this coronavirus, the efficacy of this killed vaccine might reduce significantly, right? The killed vaccine, the immunity, the type of immunity is very narrow, right? There are some other types of vaccines. One is the vector vaccines using adenovirus. I'm very curious to see these results, because you remember, in poultry, in chickens, we don't have a vector vaccine against bronchitis. We don't have, there is a technology, there are some publications, but the technology is very good to give protection against homologous challenge. If I have an insert against a Massachusetts strain, so the protection against Massachusetts is relevant, is good. But when this virus changes, the IV challenge virus changes, this type of vaccines loses efficacy very quickly. So I'm very curious to see the result of this vaccine in human beings, to see whether this could be a way to develop vaccines for poultry in the future, right? And the one that I'm very, very curious is the mRNA, the messenger, because this is completely new, and I'm really interested to learn and to see what can be expected from these new technologies. But honestly, I do believe in vaccines. I will take a vaccine for sure. I'm anxiously waiting for the vaccine, and I think this is the hope to get back to what we call a normal life.
Okay, thank you very much, Dr. Marcelo. Thank you for being with us, for giving us some time for your heavy scheduled agenda. And
my pleasure.
We also would like to thank the Ministry of Environment, Water, and Agriculture for always giving us the opportunity to be with you, to share the experience and to discuss. We are always here. If there is any question just related to any of us, and we will forward it to Dr. Marcelo and come back to you.
It will be my pleasure. And I really would like to thank you for the opportunity. It was a pleasure. And I know that Ramadan is coming, and I wish you a very happy festive season to you guys. And I hope that we can get together soon. But this time, I would like to visit you in Saudi Arabia.
You are most, Marcelo. We are most.
Thank you very much, Hassan. Thank you.
Thank you.
السلام
عليكم.
أسعد الله مساءكم. شكرا جميعا. Thank you, Mr. Marcelo. Thank you for forever. And يعني إن شاء الله hope see you soon in Saudi Arabia. أشكر شركة ماس وشركة إبراهيم البيطرية على يعني هذه الفعالية القيمة وورشة العمل القيمة عن مرض النيوكاسل. أتمنى يعني أن الجميع استفاد من هذه الفعالية والمعلومات اللي طرحت فيها. أكرر شكري وتقديري دائما يعني شركة السيفا على يعني إقامة هذه الفعالية، ويعني الخبرات اللي نقلتها إلى العاملين في المجال البيطري عن مرض النيوكاسل في الدواجن. طبعا يعني وهذا امتداد للعديد من الفعاليات اللي أقيمت بالشراكة مع الشركة في الفترات السابقة. نتمنى إن شاء الله بإذن الله أن الجميع استفاد من هذه الفعالية. لمن أراد الرجوع إلى هذه الفعالية هي موجودة على موقع الإرشاد البيطري تابع لوزارة البيئة والمياه والزراعة. أي وقت ممكن يعني يرجع لهذه المادة العلمية. والشكر لكم دكتور علاء فتوح، دكتور حازم، ودكتور أحمد حجاج ودكتور مارسيلو على يعني هذا العرض القيم والمعلومات الفنية الكبيرة يعني. فشكرا لكم جميعا.
جزيلا.
المايك معك دكتور علاء.
شكرا جزيلا دكتور علي على الاستضافة للإيفنت كله. شكرا دكتور مارسيلو، شكرا أحمد، شكرا حازم، تيم سيفا بالمملكة العربية السعودية، وشكرا لشركة ماس لاستضافتها تيم سيفا في المملكة العربية السعودية، وشكرا لكل الحضور. وشركة سيفا بتتمنى لحضراتكم جميعا رمضان مبارك عليكم وعلى الأمة الإسلامية. شكرا جزيلا.
رمضان مبارك، اشوفكم على خير ان شاء الله. السلام عليكم.